Hello, this is BiS987 Biofusion Seminar. This is Team 2, and today we are going to give you a webinar which title is Networks and Their Applications in Systems Biology.
Hello, my name is Hanjin Kim and I'll be your moderator today. And we will going to talk about networks and their applications in system biology. This webinar has a presentation session and a discussion session. In presentation sessions, Jaehyuk Choi and Yeonsu Choi will lead presentation about introduction of network biology and their applications. And then Taechan Lee, Seungyeon Wang, and Hamza Arshad Dar will compare the difference between networks used in system biology and other networks. And they will discuss the limitation and future prospects of biological networks. At first, Jaehyuk Choi will going to talk about the network science.
Okay, hello, my name is Jaehyuk Choi, under Professor Kwanghyun Cho. We study systems biology, and I am the first presenter for the second presentation of this biofusion seminar. The topic I am going to present today is about network science in general and regarding main characteristics of the networks. The first question you could ask here is why network science is being studied. In an interview in the San Jose Mercury News in January 23rd in the year 2000, Stephen Hawking said the following: "I think the next century will be the century of complexity. Today, as accumulation of data is occurring, the importance of processing such complex data is being emphasized." The definition of complex, according to dictionary.com, means to be A, either composed of many interconnected parts, or B, to be characterized by a very complicated or involved arrangement of parts. Therefore, in order to understand complex systems, we need to view the individual parts and to understand the interactions between the parts. Therefore, network science as a field began to rise and became applied in various fields. And the images over here on the right shows various examples of such networks being studied. This one over here, the top left image, shows a protein interaction network for the Alzheimer's disease. The top right over here shows an example of a human brain network. The bottom left and the bottom right each shows an example of a social media network and a power grid network used in South Korea, respectively. As shown in these examples, networks are being used to study complex systems and thus shows the importance of network science. Now, this slide explains the very basics of networks. Each components in a network are called nodes, or also known as an edge. The interactions between these nodes are called links, or also known as vertices. How many edges or how many links are connected to a node is also referred as a degree. The average degree is the total number of links. Average degrees of a system is the total number of links, all the number of the links, divided by the total number of the nodes. As you can see on the bottom left image, a network consists of 4 nodes, nodes 1, 2, 3, and 4. Node 1 has 1 link to node 2, and so therefore it has a degree of 1. And node 2 has 3 links connected to nodes 1, node 3, and node 4, and therefore has a degree of 3. Likewise, nodes 3 and 4 have 2 links respectively, and therefore has a degree of 2. This B image over here shows the frequency, the probability on the y-axis, with the x-axis being the number of degree, becoming the size of the degree. C image over here shows a network with 12 nodes, each with a degree of the size of 2. Such degree distribution of networks determine the characteristics of the system. The image on the right over here shows represents a protein-protein interaction network of yeast. Each node, each dot over here represents a protein, and the links correspond to the binding of the proteins, or the binding interactions of the proteins. The proteins over here, the bottom left here, have a self-loop and therefore correspond to a degree of 2, one that comes out from itself and the one that enters itself. These show the distributions of the degrees. You can see that most of the nodes have a degree of 1, while a few have the degree of higher degrees. And when they are plotted in a log-log plot, you can see that there are a few hub nodes, which means largely connected nodes, which will be later on explained in the slides. Networks are often distinguished by elementary properties of the graph. In this slide, I will explain some common types of networks that are used. The first case is an undirected graph. This is a case where a network where the networks have the links which do not have a defined direction. Networks such as the internet or power grids usually have an undirected graph. Another commonly seen feature in networks are self-loops. A self-loop, as you can see in nodes 1 and nodes 4, represent an interaction which a node interacts with itself. Examples include the World Wide Web or the protein-protein interaction networks in which the protein self-activates or self-regulates itself. Multigraphs, in the case of multigraphs, nodes are allowed to have multiple links. As you can see in nodes 1 and 2, there are 2 links between nodes 1 and nodes 2. And between nodes 2 and node 4, there are 3 links. Examples include social networks, such as when 2 people or 2 nodes can have multiple relationships, such as friendship, family, or professional ties, or those kind of things. The next one we're going to see is the directed networks, which in the case, the links have directions. Number 3 activates or connects with link 1, and link 1 is connected to link 2 with a direction, such as examples include mobile phone calls or citation networks. There's also something called the weighted network, in which the network has a defined weight or strength, or also known as a flow parameter. In this case, nodes 2 and nodes 4 are strongly connected, while nodes 1 and node 3 are weakly connected. The last one I'm going to explain here is a complete graph, or in this case, it's undirected. A complete graph is a network where all the nodes 1 is connected to node 3, node 2, node 3, and node 4. Likewise, node 2 is connected to node 1, node 3, and node 4. Likewise, a complete network is a network where all nodes are interconnected to each other. The next concept I will explain are random networks. Random networks are commonly used to study networks because real networks are difficult to construct and are complex. There are 2 main random networks. There is the NL model, with N being the number of nodes and L being the number of links. In the case of NP models, N equals the number of nodes, with P representing the probabilities. The image over here on the bottom side represents a complete graph with 16 nodes and 120 links. In the case of NL models, we choose L random nodes out of the 120 total links, and therefore a random network is constructed. In the case of an NP model, using the 120 links, each link has a probability P. And then using those probabilities, if generating a random number, and if it's under P, you let that node live while the other nodes are removed. Most real networks are actually scale-free networks. A scale-free network is where the degree of the nodes follow a power law distribution, like this over here. In the case of a random network, it follows the purple over here. In the case of a random network, on the other hand, follows a Poisson distribution, which is indicated in the green color. And this is a log-log graph of the same image over here. You can see that most of the nodes have similar degrees, and you can see in the Poisson distribution, while in the case of scale-free networks, some nodes are highly connected, while some nodes are very connected with small degrees. The image on the right, ABCD on the right, shows examples of real networks. A shows a network of the internet, you can see that it's a log-log graph and they have a linear distribution like the scale-free network as shown in B over here. B, on the other hand, shows a protein-protein interaction network. C shows an example of an email network, and D shows an example of a citation network. As you can see, they do not follow the Poisson distribution, represented in green, and have a linear distribution when plotted to a log-log graph. Nodes with higher degrees, or at the end over here, are known as hub nodes, which are connected to many other nodes and have a high degree. The Barabasi-Albert model is one that can generate scale-free models, scale-free networks. The Barabasi-Albert model has two characteristics: growth and preferential attachment. Growth occurs when a new node is added to an existing node. You can see when there's two nodes, another node is added, and the links are connected to the previously existing nodes. In this case, this white node is added, and two links are added to the existing nodes. Likewise, this continues on, and each time two nodes are added, which doesn't necessarily have to be two, but in this case it's two. And you can see also there's also a preferential attachment, in which case when a new node is connected to a pre-existing node, it's much more likely to connect to a node with a higher degree. In other words, larger nodes will likely acquire more links. I'll show you a brief video right here. It's 20 seconds. So as you can see, as each node is becoming attached, you can see the larger dots, which means it has a higher degree, becomes even larger, while the smaller degrees generally remain small. It shows how hub nodes are being formed and shows that the larger nodes are likely to become even larger. The final concept I would like to introduce in this presentation is network robustness of scale-free networks. In the case of scale-free networks, networks are known to be robust, or that their shape does not break down even in the presence of perturbations such as removal of nodes or removal of links. This video over here shows how when each node is being removed, you can see that most of the nodes still remain connected. The graph on the right shows each properties and shows how likely they remain connected to the large giant component until a certain fraction is reached and the network breaks down. In the case of a biological network, it shows that even if mutations are present, the rest of the system may still survive and maintain the function of the organism, eventually allowing the organism to survive despite various mutations that are present in the system. Regarding the reference of the presentation, otherwise stated, they're mainly from this book, Network Science, published by Albert Laszlo Barabasi, and it's available online for free for people who are interested. And thank you for listening to my presentation, and I wish you a great day.
Thank you for your great presentation. And Yeonsu Choi will introduce about applications of network to biology.
Okay, hello class. This is second presenter. My name is Yeonsu Choi. In this presentation, I'll introduce some examples of network biology. First of all, I'll explain how the network is applied on biology. Modern biology grows up with two properties. First, the view that biology is fundamentally informational sciences. Biological system, cell, and organism have a process which can store and transmit information basically. For example, phenotype of life is stored as genetic information, and this genetic information can be transmitted to descendant. Second, new technologies and approaches have been emerged as the time passed. These new things enable researchers studying biological complexity. especially Human Genome Project, which was conducted 1990 to 2003, became a driving force to develop high-throughput sequencing, computational and mathematical tools, which it leads researchers to analyze and understand the biological large data sets. In the midst of these changes, systems biology emerged as the study of the interactions and behavior of the components of biological entities. In aspect of systems biology, the most important thing is collective properties. It is also called emergent properties. It means how cell works result from collective behavior of many molecules. In other words, understanding only one molecular part is not enough to understand or predict system behavior. You can catch the same phrase in the citation of Pauling and Zucker-Kondo. They said in their joint paper, life is a relationship among molecules. Likewise, systems biology is the study examining the interaction among biological components, which include processes, identifying individual components, understanding role and interaction of that components, and attempting to identify how the interactions among components make biological behavior. Network is a useful tool to represent that interaction among components. Each nodes include the properties of the components, and their interactions can be interpreted as edge of the network. Here's the examples. Protein-protein interactions are gathered into protein interaction network. It is most intensively analyzed network. Protein can recognize and bind target molecule by their structure, and many proteins are used as enzymes in the cell. It can cause numerous reactions like metabolism, signaling, and gene expression. Gene regulatory network shows interaction between transcription factor and gene. Some target protein could increase or decrease some gene expression, and it could be direct or indirect. As understanding these networks, we could predict reactions from some target drug or inhibitions of some genes, and increment of some genes, and etc. Signaling pathway also could be a network. It transfers a signal from outside cells to inside the cell. Leptin is one example, which is summarized cell works occurring when signal molecule outside cell bind to receptor. Binding signal molecule to receptor modify below part of receptor, which leads continuous reactions in cell. From this process, cell adapt to altered environment. Reactions in cell depends on signal molecule and have temporal and physical complexity. Right is the metabolic map. This is network of chemical reactions for getting energy and surviving. Molecules and enzymes are included as node and edge. It represents all reaction about synthesis and decomposition. Previous researchers focused on tagging and tracking some target molecule, identifying and understanding some metabolic pathway about target molecule. Application of network in this part can make it possible to understand the complete metabolism of cell. And also ecology is another example using network in biology. Food web is well-known network that depict the circulation of molecules. Interspecies and intraspecies interactions are also network. Organisms interact within species and between species. That sea otter maintains the giant kelp forest by eating urchins is famous example for showing interaction between species. In results, network is used in many parts of biology, which make researchers enabled to understand biological phenomena, maintaining its complexity. Enormous interaction among biological entities are still actively analyzed. Network give us great point to examine and predict multiple layered effects from some targets. And this is my end of my presentation, and thank you for listening.
Okay, thank you for your great presentation. And from now on, we will have panelist discussion session. Three panelists, Haechan Lee, Seungyeon Wang, and Hamza Ashar Dar, will lead the discussion session about the difference, limitation, and future prospects of biological networks.
Hello, my name is Haechan Lee. I'm a panel for the first discussion. The topic that I'm going to discuss is different characteristics of biological networks compared to other networks. In this discussion, I'm going to explain some examples of the biological networks and other networks such as social networks. And then I'm going to explain some structural differences between them. Knowing differences between them is important because typically tools and insights from the other network analysis can be easily applied to the biological network. So social and biological networks are two major structures in network science. So in this presentation, I'm going to compare biological networks with social networks. And one example of the social network is SNS. Twitter, Facebook, Instagram are the famous SNS platforms that we usually use. In 2018, the number of active users in Facebook is about 2 billion, also 700 million for Instagram, and around 300 million for Twitter. So if we make a network using the user's information, each user can be a node of the network, and social interactions can be the edge of the network. The examples of the biological networks are protein-protein interaction networks and metabolic networks. As the other presenters said, those in PPI network represent the proteins and edges represent the physical relationship between them. So there are many PPI networks according to the model organisms. And in the human PPI networks, there are approximately 10,000 nodes. And those in the metabolic network represent metabolites and edges represent biochemical reactions between them. So in the metabolic network, there are about 100,000 nodes. So according to these examples, the first different characteristic that we can find is the size difference. And usually size of the biological network is around 1,000 times smaller than the social network. Then are there any differences in terms of the network structure? The paper in 2014 suggested that there is the structural differences in terms of network community profile. The community with in in the left figure, the community which can be called cluster means a subset of nodes within the network such that connections between the nodes are denser than connections with the rest of the network. Communities C1, C2, and C3 are densely connected internally and sparsely connected with the rest of the network. And community detection is important to analyze both networks. This is because communities can provide for a better understanding and insight on the fine-grained structure and the way their different parts work together. So right figures represent the network community profiles in the biological networks and social network. X-axis represents the number of nodes in the best cluster, and y-axis represents the conductance. The conductance value is extensively used to measure the cohesiveness of a community, and the smaller the conductance of a set of nodes, the more community-like the set is. So according to this analysis, uh, in the figure A and B, the best size of communities in biological networks is not reached at size 100 and at size about 10. However, in the figure C and D, the best size of communities in social network is above 100. From these results, we can know that the community size of the biological network is an order of magnitudes smaller than the social network. So second different characteristic is related to generative model describing the network. Real-world networks are evolving over time, either by adding or removing nodes or link over time. For examples, in social networks, people make and lose friends over time, thereby creating and destroying edges. Also, there are some generative models representing evolving networks, and according to the previous researches, the models generate networks similar to social networks and similar to biological networks are different. Social network-like graphs were generated by forest fire model, but biological network-like graphs were generated by random rewiring model. Forest fire model is the process that new edges are added via recursive burning mechanism in an epidemic-like fashion, like forest fire. On the other hand, rewiring model starts with the original network, and the model randomly select pairs of edges and switch their nodes and do this many times. So below, the figures show that the networks generated by rewiring model are similar with biological networks, but not similar with social networks. So from these results, we can know that the evolving process of each network can be somewhat different. So in summary, biological networks have their unique structural characteristics compared to social networks. Those biological networks should be treated differently, and a fine-grained analysis is needed to better understand the structure. Thank you for listening.
Good afternoon, everyone. I'm the second panelist, Singan Wang, under Professor Do Hwan Lee, and our research interest is systems biology. And I'm going to discuss about the incompleteness of biological networks in this section. As we saw in the previous presentation, network biology has been suggested promising solutions in many biological research fields, such as repurposing drugs or finding disease modules. However, it is still difficult to say yes to the question whether biological networks represent the real biological system appropriately. Hence, I want to discuss about the incompleteness of the biological networks from three perspectives: discrepancies in databases and study biases and lack of dynamic properties. First, the incompleteness of biological networks result from discrepancies in databases. In most of the cases, to construct a biological network, bioinformaticians use publicly available databases that gather the interaction data from published medical literatures. However, the problem is that there are discrepancies between these databases, even the data recorded from the same published literature. In the left figure, you can see that each database records the same number, a different number of protein-protein interaction and use different protein names from the same published literature. And in the case of protein-protein interaction network, it was labeled that only two databases fully agree on only 42% of interactions. So therefore, more standardizing curation policies are needed to construct a more accurate and precise biological network. And the second cause of the incompleteness of biological network is study biases. The study biases arises due to the fact that proteins are studied an uneven amount of times. Some proteins which have larger biomedical relevance or attract researchers' interest are studied more often than the other proteins. For example, it is well known that the disease proteins, such as cancer proteins, have a significantly higher degree in protein-protein network. So the network bias to the disease proteins can potentially influence the following research that use this kind of biological network. So we need solutions that can handle study biases between the proteins. And the last problem of incompleteness of biological networks arise due to the lack of data that can reflect the dynamic properties of actual biological systems. Actual biological interactome is a highly dynamic systems, but currently used biological networks are just snapshots of interactions and represent conceptually simplified interactions and are just the totality of interactions between biological entities. Hence, we have to reflect the dynamic properties of actual biological systems through distinct temporal and spatial and environmental data. For example, in recent years, many research reflect the dynamic properties of actual biological systems by constructing co-expression networks through spatial temporal RNA sequencing data or single-cell RNA sequencing data. So in the future, we hope that more standardizing curation policies for databases and balance it between biological entities and the data that reflect the dynamic properties can help to construct a more complete biological network that represent the real biological system appropriately. And this is my end of my discussion, and Hamza will continue the discussion. Thank you.
So hello everyone, my name is Hamza Arshad Dar, and the topic of my presentation is biological networks, the means to an end. So I will be presenting it. Now talking about the utility of biological networks, as my group members already
Excuse me, I
explained.
cannot see your screen, your presentation file. Anybody else can see that?
Actually, I had clicked on the thing, but it's not visible.
I cannot see. And TA, yeah, no, nobody can see that. Just dark screen right now. Can you actually the share again? Share screen again? You can actually stop sharing and then share again. It seems like he's disconnected, right? So his picture is just like freezed. Maybe he's reconnecting it, I guess. 저 아직 안 들어왔죠? 혹시 그 방학생 있으면 한번 다시 연락해 볼래요? 어떻게 연결됐는지? Okay. Okay.
I'm sorry, Zoom was not responding. I don't know what was the problem, so I will just continue.
Okay. Yeah. So can you share again?
Yes, I'm sharing again.
Mm-hmm.
Can you see it?
Yes, we can see now.
Okay, sorry for the interruption. So I'm going to start it now. As my group members already explained, that biological networks are very useful for the addressing questions in systems biology and disease etiology, therapeutics discovery. Now we can study both complex and common diseases through this systems biology approach, and uh, and I want to tell about that in this biological network approach, as it's part of the systems biology, so it is based on the solid theoretical foundation. What I mean with this is that uh, based on the biomedical literature and the available data, we construct a model, and then the model input is validated, and afterwards the full model is verified, and finally model output corroboration, and that is confirmation, is carried out through process and input validation. So the point I want to emphasize is that at every stage of this type of experiment, uh, there are verification and validation procedures in place, so to get the more meaningful results and to get the output in good way. So now talking about the network biology approach, as it is a part of the systems biology, so there are, needless to say, there are some constraints involved. This is constraints in systems biology, they cannot be ignored because the biological systems, they are very complex in nature, as well as there is an element of uncertainty involved. Uh, for example, due to the lack of gene expression values, uh, we can just consider the Boolean modeling. Now what is Boolean modeling? It is a type of modeling which is done on the basis of Boolean values like 0 and 1. Like you can see in the figure that this network is there, and there are corresponding Boolean symbols that represent either activation or inhibition. Now zero indicates the inhibition, and 1 indicates the activation. So based on these analysis, truth tables are constructed, and Boolean functions are applied, and finally the state transition graph is obtained. After analyzing it, the dynamics of the system, biological system, can be understood in a meaningful way. Now, what I want to emphasize is that this is a qualitative approach because that is based on the only qualitative data like activation or inhibition. There are no quantitative data involved, and due to the lack of quantitative data, we cannot infer much from this type of analysis. So, actually, the point is that in biological databases, sometimes there is lack of reliable quantitative data, which is somewhat detrimental to our analysis. So, as I mentioned that systems biology and all types of systems biology experiments, that data plays a key role. It is basically a data-driven approach, and improvements are carried out based on the data. Design of most biology systems biology studies enables continuous improvements, as we can see in this flow. That first of all, the systems biology components are defined, and then the scientists, what they do is they apply perturbations, that is either simulations, or we can check the effect of drugs, and after that we observe the results and compare the responses with the model predictions, and based on these responses and the results we obtain, new perturbations or simulations are designed, and then they are chosen, and then the process is repeated again and again until we get our desired results. So the point is that both prior biological data as well as our in silico experimentation yields some results which are important for the analysis. So studying the biological network actually helps understand the dynamic behavior of biological system, which is otherwise not possible through a traditional biochemical test, etc. So what does the future hold? As we can clearly understand that network analysis has a bigger role to play in the study of infectious diseases. So through this approach, we can just improve our overall understanding of infectious diseases. Like we can see in the figure that there's the control sample. gene expression of control sample and then the gene expression of diseased patient. So based on the differential expression of these genes, systems biology approach can be applied, and after that analysis, we can gain the enhanced understanding of diseases. Now, another thing we can do is, and we can accurately predict the function of a given protein, let's say. Another thing is we can do is we can study the biological mechanisms, because these signaling pathways, what we can do is we can study their dynamic properties, and thus we can gain useful insights into the biological mechanisms. And finally, advancements in the field of proteomics. This can also be advancements in the field of proteomics. This can be enabled by the network analysis approach. So basically, what the future hold is that network biology has is important for these disciplines: genomics, proteomics, and pathology, like understanding the disease, etc. And especially the point of therapeutics, drug discovery, because we can understand that which targets are vulnerable to inhibition, and if we inhibit the target, it will be it can be used to treat different human diseases. And so finally, metabolomics, as I already explained, that we can study the mechanism of disease through pathway analysis, and most of the time they are like metabolic pathways, so we can just study them further and apply perturbations, etc. So these are some of the aspects of these. So that concludes my presentation. Thank you for listening.
Okay, thank you for all the panelists. So this is the end of our presentations. Thank you for your attention, and feel free to ask any questions.
And you can actually ask question in the Q&A board if you can actually, or you can actually use a chat board, but yeah, you can write that. And also if you want to speak, to ask a question, you can raise your hand, then we can actually give you a mic as well. So maybe moderator or panelist read this Q&A board, and then maybe some of you answer the question. Okay.
Yeah, okay. There are some questions from chatting room. The questions from Dongyeol Seok was that, thank you for presenting the presentation about networks. The question was that I would like to ask about whether there has been any example in which system biology solved problems that could not solved by classical biology. Is there anyone who can answer this question?
I think either one of the panelists or presenter should answer this question, I think, yeah.
I'll answer for the first question.
Mm-hmm.
Regarding whether there are actually solved cases. Recently, they're actually using these network biology to find drug targets. And if I can't remember the exact name, but I remember that there was a case in which it actually found a new drug target and eventually led to a drug success. And also, if I remember correctly, our lab found other drug targets that could possibly reverse cancer and those kind of research using systems biology, which was probably unavailable using classical biology or using statistical methods. So yes, the answer would be yes, they can find novel targets, and there are examples of systems biology that could solve such issues. Did that help? I hope it did.
Okay, thank you. That was the answer for the Dongyeol Seok. And the question from Jong-Sol Kim, there are two questions from him. The first question is that I think that the introduced difference between the biological network and the social network is just an observation. Is there any study about the underlying reason?
I'm answer this question. Yes, you're right. What I explained was the one case study of the differences between the networks. The paper showed the differences using the one example, using the PPI network and social network, SNS network. So it's just on one observation, but what what I'm going to emphasize was there are some differences between the networks. So uh, I don't know exactly about the underlying reasons. It's just one case study.
Thank you. That was the answer for the first question. And the second question is that nowadays people try to apply deep learning algorithm to networks, and in many cases, I saw that they apply the same algorithm to both the social and biological network. Do you think there should be any difference in algorithm depending on the network?
I didn't research about this deep learning algorithm to network analysis, but I think there could be any differences in algorithm depending on the network because uh, 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 because the some that what I explained is the structural differences between the network. So the if we use the deep learning algorithm efficiently, then we should adapt the deep learning algorithm differently, I think. But I don't know exactly what's the differences.
Okay, thank you for your answer. Is there any more question?
Uh, I have one question actually. Yeah, actually
Yeah.
related to the second question. So yeah, nowadays many people are using like a graph neural network for the network analysis, right? Which means that there must be some area that classical graph analysis does not answer. So I'm just curious that what's actually main difference, how to say that, or maybe what's actually main weakness of the classical graph analysis compared to the recent surge of the graph neural network? Anybody can answer my question? Yeah, let me rephrase my question again. So, what do you think that graph neural network can answer? What kinds of new problem graph neural network can answer compared to the classical graph analysis? Or what do you hope graph neural network can answer? Even though you don't have any experiences, still, what kinds of, you may experience any kind some problem with the classical graph analysis, right? So that means you may wish some problem need to be solved within neural network or something like that. So is any examples of those kind of things?
Okay, I'll answer to this question. In the case of graph neural networks, actually, in the case of network modeling today, the most significant problem is actually constructing the network itself.
Mhm.
And as far as I'm concerned, I'm not sure about graph neural networks completely, but in the case of deep neural networks or other related networks using deep learning, you can't really construct model because it's mostly a black box model using, in the case of neural network systems, which sort of results in like a limitation to understanding why this sort of phenomenon actually occurred. So the main issue regarding systems biology in network analysis is actually constructing the network, not really more of a, it's more constructed, it's more focused towards constructing the network rather than analyzing the network at the current present time. So yes, there are multiple tries or attempts to use graph neural networks or deep neural networks. But at the current stage, I think that it's hard to understand why, in the case of black box models, it's hard to understand why why such decision was made. And the accuracy actually is much more, if I'm correct, it's lower compared to the the classical modeling of graphs.
Mm-hmm. So if I understand correctly, that this kind of graph construction means like a link analysis, right? Connectivity or those kinds of analysis is a big problem.
Finding whether the links actually exist between two nodes or two proteins, or whether they have a significant strength of a link, or those kinds of things.
I see, I see. So, okay. So that kind of problem is the area people are interested in looking in with the new advanced technologies, right? Okay. Great. So any other question from students before you wrap up?
So
okay, if not, yeah, you can wrap up. Yeah, moderator.
It seems all the questions are done, and so we are going to end this webinar today. Thank you for still listening.
Okay, thank you very much for all the presenters and panelists. It was, I learned a lot actually. Thank you very much for your nice preparation. Okay, great. Then we can meet you all students next time. Bye.
Thank you.
Well, thank you.